Okay, so literally hot off the press, Andy and I are going to update you about the third breakout session, and just a poll of the room, who was in the third breakout session? We had quite a packed room. Okay, so you can correct us if we've left anything out. So the theme of this breakout session was how to approach more complex features, given examples of GxE and epistasis. This is a fairly broad scope, so we spent some time actually defining what the scope of the discussion was. We split our time half and half, so half GxE, half epistasis or GxE or pathway analyses. We discussed these processes both in humans and model organisms. We explored the scope in terms of cellular or tissue level all the way to population-based applications and applications into health systems. We thought about it both in terms of basic research and also in terms of implementation science and genomic medicine, and how this would then link to other types of experiments, like the kind of perturbation we heard about this morning. We identified some of the gaps in data. There was a feeling in the room that the time is now for some of the data for this type of multi-omic exploration, that now is when a lot of data types are coming online at scale. And we debated some of the study design and some of the models that we would think about using. For example, models focused on a sort of forward engineering approach in specific cell types versus collecting data on many types of data on many types of people. We also thought about why these attributes and exploring these complex features was NHGRI's job, and we came up with some rationale. Fundamentally, this focuses on functional aspects of the genome as a primary goal. This is basic biology, and traverses many diseases. In fact, these attributes can be shared across different types of diseases. So the role of NHGRI here would be very complementary to the disease institutes. NHGRI is positioned to really scale up targeted studies if they work and if the design works, and this is seen as a positive. And we thought it was the next logical step after understanding, in quotes, single allelic effects on the genome.
All right. So we did jump around a little bit between specific sort of design ideas and talking about very general issues of how one makes inference about gene-gene interaction and GxE. These are problems that, of course, suffer from this issue of dimension explosion, that immediately there's way too much to handle. And so how does one restrict the problem to be able to make progress? So with respect to GxE, the first problem everybody faces is we don't measure the environment very well. So how might the NHGRI encourage technologies and approaches to measuring environment in a way that's meaningful for this particular problem? So we kicked around ideas such as integration of wearable devices, environmental surveys, linking up specifically with NIEHS with projects that deal with environmental impacts on disease and risks. And so this first one is just this grand sort of problem of linking up all these environmental effects with genetic variation data. And because of this dimension explosion, of course, it's going to put a huge burden on the computational problem. So are there clever ways to do feature selection to minimize, to reduce the dimensionality? Machine learning approaches and so forth is going to be at a real premium in this particular problem. The idea of integrating familial lifestyle and environmental exposures in a polygenic risk score-like model, where instead of just the genotype of the individual at birth, we also integrate some of this information on the environment, would be a very desirable goal. One immediately has to get into the details of how those models would be designed, but it's an interesting problem. One particular idea that was floated was we sort of threw up our hands and said, well, this is just too much. We're not going to be able to make progress at such high dimension. Let's instead focus on a few very much more focused problems of specific disease phenotype, specific restricted set of gene regulatory networks, and really dissect them in much greater detail. So that was one idea that was floated. It's not like we voted on these, but that was one that obviously we could make progress that way. As we've collected this information on many, many individuals, we identify not only single SNP variants, but sort of haplotypes for whole regions of the genome that are particularly known to be of large functional significance to particular phenotypes or diseases. So these different haplotypes represent a sort of allelic series, if you will, in the sense of model organisms. And so across individuals, we have many, many different genotypes of those combinations of those allelic series. And this, too, provides a means for dissecting all the effects of the transregulatory factors that hit that part of the genome and provide a particularly nice molecular dissection of that particular gene or gene region. This idea of causal inference in the sense of Judea Pearl's book on causality, and he has another one more recently, the statistical inference of causation of either genotype, phenotype, or relationships between genes through either temporal or lagged correlations or through partial correlation. There are several ideas floating around in the high dimension statistics world for inference of causality. And these are being underapplied in the context of inference of gene-gene interaction. And there are a lot of opportunities for doing that, so we encourage that. Evolution came up in our section as well, and of course epistasis was of considerable interest to many evolutionary biologists. So the role of gene-gene interaction, or as Richard Lewontin put it, context is of the essence, in the way that the allele frequency dynamics of a gene are impacted by other genes is, of course, critical in evolution. So how is this relevant to the human condition? How is it relevant to particularly the situation where we have in humans a very unusual genomic situation of a rapidly expanding population giving us lots of very rare alleles, potentially of larger effect, potentially a lot more epistasis, in fact, in humans for that reason. Study of gene regulatory networks across disparate populations is also something we'd like to see happen, where we can actually ask about the stability of those inferred interactions. Do all populations show the same nature and amount of such interactions, and do we expect that? And finally, a particular sharp, crystal clear, really wonderful example of gene-gene interaction was particularly illustrated by Rick Lifton's work, where we have these very large effect Mendelian disorders, where when you study Mendelian disorders at scale, you find individuals who are homozygous for loss of function mutations, but are perfectly normal. So you then augur in with those, and Lifton did it by sequencing. You identify then, in some cases, second site suppressors of that phenotype. So this is a very, very nice design. Does it scale up better for identifying large effect alleles that are directly interacting either positively or, in this case, of course, negatively in the sense of correcting the phenotype? So that's it. I'm sure we went over.